Hello, everyone, and welcome to the Emerging Technologies Information Sessions 2025 webinar series, week 4. My name is Sam Semino, and I work for the USGS and the Pacific Northwest Aquatic Monitoring Partnership. And over the last year or so, a planning committee has been putting together this ETIS webinar series, but we didn't start from a clean slate. The Pacific Northwest Aquatic Monitoring Partnership and the Pacific States Marine Fisheries Commission's StreamNet program have been hosting various versions of these emerging technologies conferences and webinars for more than 14 years now. And the overarching goal of the Emerging Tech Project is to bring together monitoring professionals, project managers, and data managers to showcase how they implement the latest technologies or old technologies in new, novel ways. We are in our 4th week of this 5-week webinar series, and with our final session being next week, Wednesday, February 5th, and it'll be from 9 a.m. to 11 a.m. Pacific time, or wherever you are, adjust accordingly. And today's session, we are covering genetics, and we will have 4 presentations on the advancement of genetics and various aquatic monitoring practices. These sessions are all being recorded, so if you missed any of the first 3 sessions, you can check out those recordings on the PNAMP YouTube channel. You can search YouTube for the session names, and usually you need to search for PNAMP, PNAMP. And these will typically be available within a week of each of these sessions. So look for it tomorrow or Friday. And now I'm going to kick it over to today's moderator, Alan Martin.
Thank you, Sam. Good morning, everyone. My name is Alan Martin. I'm a project manager for Pacific States Marine Fisheries Commission. Thanks for joining us today for the 4th session of the Emerging Technologies Information Session webinar series. As Sam mentioned, we have 4 30-minute presentations on genetics today. So before we get started, I'd like to go over a few housekeeping items. On your screen, you should see some of the commonly used icons in Teams. Unless you are speaking, please turn off your camera and microphone. If you have questions or feedback, please use the chat function or raise your hand and unmute yourself. We can mute you, but we cannot unmute you. We are recording the meeting, and it will be available on the PNAP YouTube channel. A couple additional troubleshooting tips. If you're having trouble with functionality, try leaving and joining again. You can also try using a different browser or try using the desktop version of Teams. If you don't have access to chat, please feel free to unmute yourself or and to comment or ask questions. And next slide. Okay, so a lot of verbiage on here, but the takeaway is PNAP commits to fostering an open and welcome environment free of harassment and discrimination. So please remain respectful. All right, so let's begin. Just a note, after each presentation, we have about 5 to 10 minutes allotted for questions, depending on time. And if we have extra time at the end of all the presentations, we'll open the floor again for questions on any of the presentations given today. I'm going to hand this over to Kelly, but while she's getting set up, it's my pleasure to introduce her. Dr. Kellie Carim is an aquatic research ecologist with expertise in using genetic tools and information to address questions related to conservation of freshwater biodiversity. She earned a BA in biology from Carleton College, followed by a PhD in fish and wildlife biology from the University of Montana. Prior to joining the U.S. Forest Service Aldo Leopold Wilderness Research Institute in 2021, Kellie worked for 7 years as a research biologist with the U.S. Forest Service National Genomic Center for Wildlife and Fish Conservation. Here, she focused on developing and applying eDNA methods for stream settings. However, her research experience is broad and includes work on habitat suitability, connectivity, genetic diversity, phylogenetics, and life history dynamics of native and invasive aquatic species. Today, she will share some of her recent work examining current taxonomic diversity of lampreys, the oldest group of fish on Earth. And with that, Dr. Kellie Carim.
Great, thanks, Alan. All right, so I'm just going to kick it off and ask, what are lamprey? A lot of folks are not familiar with this fish, and I've heard people wonder if they are reptiles or amphibians, but they are not. They are an ancient lineage of jawless fish. And in fact, from an evolutionary perspective, lampreys are the original lineage of fish, meaning that all other fish evolved from lampreys. They're also one of the original vertebrates. So in a way, all of us evolved from lampreys. Now, we have estimated that lampreys have been on Earth for about 350 million years, which means that they predate dinosaurs, trees, and even the rings on Saturn. And another interesting fact about lamprey is that they are largely cartilaginous, and teeth are the only bony structures in their bodies. Okay, so aside from these super cool facts about lamprey, why should you actually care about them? Well, many of you may be familiar with lamprey because of the impacts invasive sea lamprey have had on native fish in the Great Lakes area. But this is just one of over 40 recognized species of lamprey worldwide, including several native lampreys in the Great Lakes area that get a bad rap from the sea lamprey invasion in that region. And throughout history, lampreys have been incorporated into culture at a global scale. And so, for example, sea lamprey have been a European delicacy since medieval times. Here I'm showing you a sea lamprey pie, where sea lampreys are native in Europe. They have been a delicacy, particularly in the United Kingdom, where these sea lamprey pies were historically served at events like coronations. And in North America, native lampreys are a very important subsistence food and integral to the identities of many Indigenous people. And so here I'm showing you a Pacific lamprey that is filleted in the traditional manner that the Yakama Nations fillets them for preservation, so that they have subsistence later in the year. And from an ecological perspective, native lampreys also play important roles in the ecosystem, so serving as a source of food for other animals, as well as filtering clean water and transporting nutrients across ecosystems. And today I'm going to talk to you about classification of lampreys with a focus on the lower levels of classification shown here. So we have 3 families of lamprey that are distributed, with 1 that's found in the Northern Hemisphere and 2 in the Southern Hemisphere. Now, across this global distribution, there are 10 recognized genera and over 40 recognized species, as I previously mentioned. And here I'm showing you 3 examples that highlight the phenotypic diversity of lampreys across the globe. And as with most fishes, this broad diversity of lamprey has historically been categorized into species based on adult morphology, and specifically the oral discs that I'm showing you here, so their teeth, as well as things like life history, variation, and feeding behavior. However, there's increasing availability, with the increasing availability of genetic information and analysis, there's mounting evidence that some of these classifications based on phenotype are inaccurate. And one group where these inaccuracies are apparent is in the genus Lampetra. This genus is a diverse genus with species that occur throughout North America and Eurasia. And the species that I'm showing you here are those that are currently recognized based on morphological classification or morphological characteristics, with the exception of 2, Lampetra erzii and Lampetra hubbsii. So let's start with Lampetra hubbsii. This is also known as Kernbrook lamprey. And when this species was first described, it was placed in the genus Entisphenus based on the dentition. And this table is showing you the criteria that Vladiakov and Kott used in 1976 to describe Kernbrook lamprey as a member of the genus Entisphenus. However, with increasing genetic information, we've performed genetic analysis and found that Kernbrook lamprey are much more closely related to the Lampetra of Western North America. And so they are now accepted as a part of this genus. Now, in contrast, we have examples of fish that look different but are actually the same. So, for example, Western Brook and Western River lamprey, these fish have long been categorized as separate species based on adult morphology, feeding habits, and life history traits. However, genetic analysis has shown that these are genetically indistinguishable at the species level, and they more accurately represent life history variants of a single species, Lampetra airsii. And this type of variation in fish is not uncommon. A very common example that you may all be familiar with is steelhead and red band rainbow trout are both considered Oncorhynchus micus. Now, beyond these few examples, there are additional genetic studies demonstrating further inconsistencies in the classification of Lampetra. For example, there's evidence that the Lampetra across these three regions may represent distinct or separate genera, and that there's likely additional species that are not being recognized within these groups. But it's important to know that these inconsistencies in classification were not formally tested by the previous studies, in part due because these previous studies were not focused on testing these hypotheses. These were more sort of interesting results that emerged from the interpretation of these previous studies. And in addition, these previous studies just had smaller data sets that were limited in sample size and geographic scope, and so they weren't able to comprehensively address these inconsistencies. But clarifying these inconsistencies is more than just a practice in helping us place fish into categories. It actually has some important management implications. So, for example, a little over 20 years ago, Lampetra of Western North America were petitioned for listing under the Endangered Species Act. And although the petition was denied due to insufficient information, it's important to understand that identifying the units for listing and recovery would require an understanding of genetic diversity both within and among species. And this sort of lack of information is part of that underlying denial of the petition, is that they just didn't, the Fish and Wildlife Service didn't have enough information to really categorize what recovery and decline looked like for the species group, for these species groups. Now, even without ESA listing, states are defining management actions and policy based on the current list of recognized species. And so if we don't understand population boundaries or even the differences between species, then prioritizing those units for conservation and management becomes very difficult. Now, to address these issues, we performed a phylogenetic analysis with a focus on Lampetra of Western North America. And we focused on two study questions. One, do the Lamprey of Western North America represent a distinct genus? And how many species exist within the Lampetra of Western North America? All right, so when classifying organisms based on phylogenetic information, we defined a genus or a species as a group with reciprocal monophyly among lineages. Or in plain language, what we're saying is we want to see all individuals of a given genus or species grouped together in a phylogenetic tree, and we don't want that group to include fish from any other genera or species. And so basically, we want to create a mutually exclusive group. So to address our first question, we used publicly available sequence data for Northern Hemisphere lamprey species at 2 mitochondrial genes and estimated maximum likelihood trees and looked at genetic distance among genus level groups. Now, we did this for Site B and COI, but the results were the same for both genes. And so I'm only going to show you the Site B results just to simplify. Here I'm showing you a phylogenetic tree that shows the groupings in our data set that have been collapsed down to the genus level. And these numbers here are bootstrap values that are showing support for the branching pattern that you see. And I'm only showing bootstrap values over 85%, which is considered very strong statistical support. Now, based on this tree, we're seeing very strong support for classification of several genera based on reciprocal monophyly. So for example, let's take a look at Ichthyomyzon. All of the specimens in this branch are identified as Ichthyomyzon, and all specimens that have been identified as Ichthyomyzon are in this branch. However, when we look at Lampetra, we're seeing an example of a polyphyletic relationship, and the Lampetra are not grouping together in the same branch. Now, taking this a step further, when we compared the genetic sequences, we found that the group from Western North America was a minimum of 6.3% different from the other groups of Lampetra, and that is fairly substantial. Now, what these results are indicating is that recognizing the group from Western North America as a separate genus is more accurate. And while I didn't show you all the details, our results also support recognizing the Eastern North American group as its own genus. However, we're not focusing on that today. So with this group from Western North America, we are formally reassigning it to a new name, Occidentis. And Occidentis is Latin, and that simply means Occidentis simply means westerly or of the West, which reflects the very broad geographic range of this taxonomic group across Western North America. And for those of you who are not sure, I want to just confirm that yes, this is a Prince reference in this image. All right, so moving on to our second objective, we were interested in understanding the species diversity within this newly named genus. And to do this, we performed a species delimitation analysis. And we drew upon several previous studies to do so. So first, Boguski et al. This study provided substantial information on Occidentis from Alaska to California, and it also identified several lineages that may represent undescribed species. However, this study didn't perform a formal species delimitation analysis to identify candidate species. The next study, Carim et al., drew heavily upon previous studies, including the Boguski study, and did perform a formal species delimitation analysis. In addition, they also included new data from Oregon as well. And one of the limitations of this data set is there was not very much information available for California at the time. But at the same time, Oringer et al. Was also working on a study performing species delimitation analysis with a focus on California. And that study came out at about the same time. And so what we wanted to do is combine the information from all of these studies, as well as several other smaller studies, to get a more comprehensive assessment of species diversity within Occidentis. This resulted in a data set of nearly 200 sequences that we examined at the site B gene. And to assess the species diversity, we used 5 different methods for species delimitation. These methods each have unique limitations or different limitations from one another. And so we drew conclusions based on the most consistent results among the methods. Now, to display the methods, I'm going to show you the maximum likelihood tree because that's really the easiest sort of visual to digest. So here's our maximum likelihood tree for Occidentis, and unlike the genus level tree I showed you earlier, I have not collapsed any of the branches into groups. So here each leaf represents a unique haplotype observed in the data set. Now in this data set, we found support for 10 candidate species. Let's walk through those. 3 of those candidate species are those that are already recognized species. We also saw support for 4 candidate species that were in agreement with species identified in Carim et al. And I want to note that these are the lineages that Boguski paper identified as possible species, but they had not performed that formal species delimitation analysis to kind of demonstrate their genetic distinctiveness. Now, 2 of these, as you'll notice, are from California, and they were also included in the study conducted by Oringer et al. Oringer et al. Found that these 2 also were distinct species in their analysis, and they also identified 2 additional lineages as species from Alameda and Napa Creeks. All right, so now we have 9 species that are in agreement with previous studies. The final and 10th candidate species identified in this study is a bit more open to interpretation, and it was represented by a single fish sampled from Paynes Creek in California. Now, it's a bit confusing because this fish was identified as a candidate species by Oringer et al., but the analysis by Carim et al. Grouped this fish with Occidentis hubbsi. So which is it, and how do we interpret these sort of varying results across studies? Let's take a closer look. So in our data set, Occidentis hubbsi and the lineage from Paynes Creek were observed in different river basins separated by several hundred river kilometers. However, they show minimum genetic divergence. So the sequences we examined were only approximately 1.3% different, which is less than the 2% threshold that's typically considered for species level differentiation. However, this sample, the sample size for both of these is extremely limited. So again, that Paynes Creek sample, that Paynes Creek sequence is represented by just a single fish. And the Occidentis hubbsi sequences are represented by 8 fish that all shared an identical genetic sequence in this data set. So we really need more information across these river basins to understand the grouping of these lineages and how they're related. Now, to highlight the importance of more sampling and what can happen if we had a smaller data set, let's take a closer look at Occidentis airzii. This species had the largest geographic range and also showed the greatest genetic diversity, with samples from over 130 locations representing 49 unique haplotypes. However, had we only examined a subset of these lineages, we likely would have come up with a different number of candidate species in our analysis. And so to help visualize this, I'm going to show you these data in the statistical parsimony network. Now, a statistical parsimony network, if you're not familiar, is just another way to visualize how similar or different gene variants or haplotypes are to one another. Each node represents a haplotype, and each segment represents a single base pair difference between the haplotypes in the data set. All right, so here is our network for Occidentis airzii, and what gets really interesting with this network is when we look at where these haplotypes fall across space. So some of these gene variants, like this one, were observed in many locations. In particular, this gene variant was observed in 19 subbasins from Alaska to California. However, many haplotypes were found in just a single river basin, creating some high levels of genetic structure. So for example, this particular haplotype was observed only in the Mackenzie River in Oregon. And this haplotype was observed in only one location in the Sacramento River in California. And these two groups represent some of the most highly diverged within this network. The genetic sequences we examined from these two differ by 2.3%. And as you recall, I said that about 2% divergence is what's typical for species level differences. So had we examined these two alone, we may have concluded that they're separate species. But the reason they didn't come out as separate species in the analysis is because we were able to put them in the context of so many other gene variants that them in this network. And this pattern is what's indicative of lineages that are likely in the early process of speciation, but they haven't quite diverged into those separate species yet. All right, so let's zoom back and talk about all of the distribution of all the candidate species together. Occidentis airzii was the most widespread, while many other candidate species were observed in just a single river basin or a single subbasin. And this could be in part because we didn't have as much sampling in southerly latitudes. However, given the history of glaciation that primarily occupied the northern regions here, we generally actually expect this type of pattern where we see more species diversity and higher rates of endemism the further we move south. And so we suspect that there's more unrecognized species, likely in coastal basins and southerly basins, more southern basins in this geographic range. So this highlights the need for more range-wide sampling of Occidentis. Even with this a broad spatial distribution in our current data set, the data set was not generated with systematic sampling. And it's clear that we have a lot of gaps in the spatial distribution that's represented here. So we are making a call for systematic sampling to first understand species diversity and geographic range of those species, and then to clarify some of these more detailed circumstances. So clarify the placement of Occidentis with respect to Paynes Creek. So is that Paynes Creek sample its own lineage or own species, or is it part of the Occidentis hubsii species? And we also expect this to improve our understanding of the genetic structure of the Occidentis airzii species complex. All right, so just to summarize, brook and river lamprey of Western North America have been reassigned to their own genus, Occidentis. And there's greater species diversity within this genus than is currently being recognized. However, I want to highlight that these candidate species are hypotheses, and the number of candidate species that we recognize is likely to change as we gain more information. And it's also important to note that the formal species descriptions require detailed information on both morphology and geographic range. And so what we're providing here is really just that first step of demonstrating the genetic distinctiveness. And we have a number of these hypotheses to chase down to solidify and really describe these as independent species. So with that is another plug for the systematic sampling of this genus, and we are undertaking some work to start addressing this. So in an effort to begin this systematic sampling, we're working with partners to determine the distribution of Occidentis species using eDNA and to develop occupancy models to understand where locations where this genus may be present. Now, based on the current funding, these efforts have been focused primarily in Oregon and Washington. But we are looking for opportunities to expand. So if this is something that interests you, please reach out. Now, the next step, once we know where this genus occurs, is to conduct sampling of the fish themselves to collect tissue samples for phylogenetic analysis and get a better understanding of morphology and really get at some of these deeper levels of species diversity. And if you'd like more information, we have several papers that can provide a few more details. This includes information that I was unable to share with you today, including fine scale population structure of Occidentis airzii observed in the Willamette Valley in Oregon, as well as some interesting sort of patterns that we suspect have kind of linking the history of glaciation to where we see different lineages of Occidentis airzii and how glaciation has shaped genetic structure on the landscape. And with that, that concludes everything I had to share with you today, and I'd be happy to take questions.
Thank you for that great presentation, Kelly, and I do appreciate the Prince reference. That's awesome.
Great.
Okay, so we'll open up the floor for discussion. It doesn't look like there's any questions in the chat, so we'll just take questions from the audience.
Hey, Kelly, this is Sam. Great presentation. I was wondering if there's any chance that some hybridization is potentially going on and how that might muddle some of these signatures or anything like that.
Yeah, that is a great question. So, you know, I think the first thing is that we don't know if hybridization is happening because we don't have enough data to investigate that. We are looking at mitochondrial DNA, which is the maternal lineage, and that should help at least at that level kind of parse that apart, because it's not a nuclear analysis would include DNA from both parents. And if you were working with a hybrid, that could definitely muddle results. But, you know, I will say, if I'm just kind of speculating, lamprey are not a fish that have been moved around as much as we might anticipate with other species like trout. And so I would be surprised if we saw a lot of hybridization. And in fact, a lot of lamprey have been targeted for removals because in past management, we kind of thought that anything that wasn't a trout or a salmon was kind of competing and taking away from trout and salmon. So I hope that as we investigate this further and suspect we probably won't see a lot of unnatural hybridization, I would say, as far as like species being moved around. But where, you know, we have species coexisting, I think it's sympatric species, it's probably very likely that we would see hybrids, and we would need genomic information to really suss that out on the landscape and see, okay, where they co-occur, what are the genetic patterns, do we see them interbreeding, and when we have subbasins where they're not co-occurring, what do those lineages look like?
Great, thanks.
All right, well, thank you, Kelly, for that great presentation. You're going to stick around for questions at the end?
Yes, I will.
Thank you, Kelly. All right, so next up we have John Hess. So as with Kelly, I'll give you a background on John and then hand it over for his presentation on lamprey restoration. Dr. John Hess is a senior fisheries geneticist and has worked with the Columbia River Intertribal Fish Commission, also known as CRIPFIC, since 2009, mostly as a geneticist in the fisheries science department, but he spent 3 of the last 16 years in fisheries management. His research focus is on population genetics of salmonids and lampreys of the Columbia River and applying genetic methods to aid fish restoration. Prior to that, he accomplished his PhD from Washington University in St. Louis and worked for 5 years as a postdoctoral scholar at NOAA Fisheries in Seattle. And with that, Dr. John Hess.
Thank you very much, Alan. And it's great to see so much representation of lampreys in this webinar. Thanks for Kellie's great introduction to lampreys in general and all the context that will be useful for my talk. So yeah, my talk is titled, How Might Restoration Efforts Help Bring a Bumper Return of Pacific Lamprey to the Columbia River? And, you know, I like to begin my talks telling you what the punchline is, so, you know, folks don't feel like they have to pay attention to all the details in this talk. And in this case, you know, the main takeaway is that we have a basic model that predicts that 2027 could be a really fantastic, hugely fantastic year for Snake River origin Pacific lamprey, and in fact, more generally, the run at large of Pacific lamprey in the Columbia River as measured at Bonneville Dam. And you will have to pay attention to the rest of the talk if you want to know the details behind why we say this. I wanted to show off my abilities in AI with this text-to-image generator depicting what it might look like if hundreds of thousands of Pacific lamprey showed up at Bonneville Dam in one day and piled up at the base of the dam. The cryptic member tribes are working hard at restoration activities aimed at making this a reality one day. So for the major results that I'll be talking about, first, you know, we have the ability to precisely determine natal origins of Pacific lamprey across a large geographic area. And this is for applications for monitoring the tribal restoration activities in the Columbia River Basin. Second, we've been seeing that juvenile production has been increasing as measured in the Snake River at Lower Granite Dam, and that these juveniles can leave the Columbia River, go out to the ocean, and we actually see Snake River origin adults return to Bonneville Dam. And in fact, we also have been seeing the offspring of translocations from the Snake River return to Bonneville Dam as adults as well. And then finally, we have a model that's predicting that the run at Bonneville Dam may reach a 10-year high in the near future. So most of these results have been published in this journal article titled The Return of the Adult Pacific Lamprey Offspring from Translocations to the Columbia River. So first, how do we have this ability to precisely determine natal origins of Pacific lamprey? Well, we have two major tools or resources that we depend on for this. And the first is a very large SIB-SHIP baseline. And what I mean by that is we've taken over 59,000 larvae and juveniles across the Pacific lamprey species range, not only the Columbia River, but outside of the Columbia River. And we use these collections of larvae and juveniles to assign the brothers and sisters of these fish at any life stage all across the range. And this helps us identify the precise natal origins of areas where fish have been translocated, but also areas where they're volitionally produced. And what I mean by volitional production is when a fish volitionally, by its own volition, gets to a spawning area and spawns without the help of humans. So next, we also have a very large parentage-based tagging baseline. We have almost 27,000 fish in this baseline, and this is comprised of all the adults that the three cryptic member tribes that participate in translocations have released in their seeded areas, and that includes Yakima Nation, Confederated Tribes of the Umatilla Indian Reservation, and Nez Perce Tribe. And these fish have been released over the years from 2007 to 2022, which is all the fish that are included in this baseline. And so it includes quite a lot of fish over the decades. And then we have an informative set of markers, or single nucleotide polymorphisms, SNPs. We have 259 of these, and these informative markers really help nail down these precise natal origins by allowing us to accurately assign these fish, whether it's sibship or parentage, to these different baselines. And then we have this large collection of unknown origin adults, and they're collected from all across the species range. But for this talk, I'm going to be narrowly focused on the ones collected at Bonneville Dam, collected between 2019 and 2022. And with those fish, we're going to use those baselines to get the precise natal origins. So the next part of this is we wanted to have an estimate for what juvenile production was behaving like in the Columbia River Basin. And for that, we looked at the Snake River juvenile production, primarily at Lower Granite Dam in the Snake River. And we had to turn to the Fish Passage Center's Brandon Chalkley, who gave us a data set for samples over our study period. And using sample rates, we expanded those samples into collection numbers and estimated juvenile abundance across these years from 2011 all the way to 2018. And what we saw is that these estimated abundances varied quite a bit, in that some years like 2015, they were quite low, only 1,000 fish estimated. And then in 2018, we saw a record year of almost 51,000 juveniles recorded that year. So since then, this publication, we've added and extended the number of years after 2018. So now our data set includes 2019, 20, 21, and 22. And with this new set of data, we see a new winner for the ultimate abundance over this time period, almost 379,000 in 2022, which is a 7-fold increase from our prior winner in 2018. So next we wanted to ask, what happens to these juvenile Pacific lamprey when they leave the Columbia River and go out into the ocean? Do they ever come back to the Columbia River? And specifically, do these Snake River fish from Lower Granite Dam that we've estimated as juveniles, when they leave Lower Granite Dam, do they ever make it back to Bonneville Dam as adults? And in fact, they do. So we've shown in this figure one cohort year of juveniles estimated in 2011 to be over 10,000 juveniles that year. And they left the Lower Granite in a single pulse. And then 4 years later, 250 of their brothers and sisters show up as adults at Bonneville Dam. And the next year, 2,500 of them show up, and then 3,000. And then 2018, there were 750. That's 7 years later after they left as juveniles from Lower Granite Dam. So it's quite amazing find. From this data, we can estimate ocean residence time. And the way that we do that is we take the difference between the peak when the juveniles left Lower Granite to the peak when they returned, that same cohort returned as adults to Bonneville Dam. And so in this case, we would get an estimate of an average of 5.5 ocean years since the time that they left as juveniles and returned as adults to freshwater. And then with all this data, looking at juvenile production and the trends in juvenile production, the ocean residence time between these cohorts, we can build a simple model that we call juvenile to adult return, or JAR. And using JAR, that's just taking a calculation of the number of estimated juveniles in a particular cohort. So in this case, 10,585 from that 2011 cohort, and dividing it by the number of adults that we've estimated from that cohort to return to Bonneville Dam. And so in this case, we had almost 6,700 adults, and we get a ratio of about 1.5. So for every 1.5 juveniles that left Lower Granite in 2011, 1 adult returned to Bonneville Dam. The salmonid biologists would be salivating over these survival estimates, but I just wanted to make the caveat that this is likely not a reflection of the true JAR, which would be a survival estimate, but it's really a proxy that's been beneficial for making a model. I think the true JAR would be much lower than this. Okay, so next, we not only have information on the volitionally produced offspring, but we also have information on the translocation offspring. And so this is showing from 2011 to 2022 how the percentage of translocation offspring has changed over time. It's increased from almost 0% to about 19% by 2018. And then after 2018, the percentage hasn't increased, but the overall numbers of translocation offspring have increased up to 50,000 that were estimated in 2022. And then we can take this information, apply the JAR model, and show when or how much of these estimates of juvenile production for the translocation fish, how much that would be estimated to translate to in terms of adults at Bonneville Dam in future years and the timing of that. And so we can see that the first little blip in 2014 comes back as adults primarily in 2019, followed by the portion that came or left Lower Granite in 2016 as juveniles. It comes back in 2022 and on until we get to the last big year, 50,000 translocation offspring leaving as juveniles from Lower Granite are predicted to come back in a large pulse in 2029, almost 3,000 adults that year. So we can take all this predicted adult abundance and sum them up, which is the dotted black line there. And from this, we estimate that in 2021, this model would predict to hit a total of about 1,100 Snake River translocation adult offspring at Bonneville. And then by 2027, we would hit nearly 5,000 adult offspring from those translocations at Bonneville. So this is all good and well. We have this model that's predicting great numbers, but we also have some real information on recent years at Bonneville where we've sampled adults and we've looked to see how many translocation offspring they represent. And in fact, in 2020, we detected the very first translocation offspring to return to Bonneville Dam as an adult. And in 2021, we were able to take as many as 3,200 adults as a sample from Bonneville and estimate all the natal origins that they represented, including the translocation programs. That year, the run at large at Bonneville was estimated to be 83,980 in 2021. We estimated that among the translocation programs for the member tribes, the translocation adults amounted to an estimated 1,065 adults. And keep in mind, our model predicted that in this year, just from the Snake River alone, that we would have nearly 1,100 just from the Snake River. The model predicted that. What we actually found was about 400 Snake River adults that year. And then we've been focused on the Nez Perce tribes and the Umatilla tribes translocation programs in the Snake River because we have a lot of data on that. But all three of these translocation programs from the member tribes have been sampled 100% since 2013, which has allowed us to estimate the numbers for the other tribes. And then we also have from the Warm Springs tribe, they've been monitoring volitional production of juveniles from 15 Mile Creek. And so we have that estimate included, the volitional production from that area as well. So moving on to 2022, we have another year of data where we've sampled 1,300 fish from Bonneville that year. The run size in 2022 at Bonneville was estimated to be about 114,000 Pacific lamprey adults. And then we've estimated all the precise natal origins, including for the translocation programs, they were estimated to return 2,200 adults that year. And then keep in mind that from the Snake River alone, the model projected about 1,600 would return that year, and we found that in reality, about 900 fish came back from the Snake River programs. And again, Warm Springs volitional production was estimated there, about 500 adults. So now let's go back to the model outputs. These are all the predictions of not only the translocation offspring, but all the Snake River origin fish in total over the years, how much they would come back to Bonneville. And we have from 2014 through 2022, these were actually estimates that we observed at Bonneville. And then from 2023 onwards, these are the model's estimated numbers for each of these cohorts. And in the gray line here, we have the total adult abundance estimated at Bonneville through 2023. And so what we looked at here is how closely the total abundances of adults at Bonneville reflected the trend in abundances from the Snake River origin fish alone. And using that ratio, as long as that stayed consistent, we were able to project out how Snake River origin abundance would translate to the run at large at Bonneville. And this led to the model showing that by 2027, we could get a record year of 450,000 fish if this model held true. Now, I want to point out that the final numbers came back from 2024, and those final estimates were a run size in total at Bonneville of about 91,000, which was more like a third of what the model predicted. So, don't come complaining to me if the model doesn't work out for 2027. But the important note here is that the underlying numbers in the model are based on juvenile recruitment. And the juvenile production from the Snake River has been extremely strong in recent years. And so as long as the ocean conditions cooperate, we could get this bumper return by 2027. And this would really help restoration of Pacific lamprey. So I just want to end with a bunch of acknowledgments. Of course, all the CRIPFIC member tribes, Pacific lamprey programs, have a very talented group of staff who are responsible for generating all this great information. And the Hagerman Lab runs all these samples required to allow us to do these tens of thousands of fish in each of the SIP and parentage baselines. We get our funding from Bonneville Power Administration, and Jean-Michel Basquiat is the artist that I use for emphasis in this presentation with those crowns. And with that, I'll take any questions that you may have.
All right, thanks for that great presentation, John. Okay, so we'll open up the floor for any questions that anyone has for John. Let's see, I've got a question. We'll start off with Doreen McCoy from the chat. So Doreen writes, curious as to where the translocation location was in southern Idaho, NO2. What are the permit requirements for translocation and transfer of species across state boundaries?
Yeah, good questions. I believe the location that you were referring to in southern Idaho was kind of an experimental or educational release in the Boise River for a class that the Nez Perce tribe was working with, I think down here. So not typical to be released above the Hells Canyon Dam complex. And then the second question was about permitting for crossing state boundaries. And with the tribes, they have pretty good flexibility for translocating fish from Bonneville, the Dalles, and John Day to the upper reaches in the Columbia River Basin, not just tied to state boundaries, but part of their ceded areas that cross state boundaries. So I think that permitting is, largely provided by the tribes.
Okay, any additional questions for John?
Hey, John, this is Mari Williams. You spoke of the model predicting historical returns. Could you put that, I forget your phrasing, sorry. Could you put that beside historical run estimates? Like that 450,000 number would be a lot compared to what we have now. Do you have an estimate of historical?
Yeah, so, we do have estimates of historical numbers prior to when the main stem dams were built. But these are pretty hazy estimates, but those estimated historical numbers would definitely dwarf an estimate of 450,000 fish. They were in the millions. And you should see a picture of Willamette Falls. It actually, the picture of Willamette Falls was what helped drive this AI image, because, Willamette Falls used to be coated with like a thick Medusa hair of Pacific lamprey in the past, and it was just in the millions of fish. And that's kind of what you would see on a typical year. So, 450,000 wouldn't be able to touch some of those historical years, but it would definitely be getting us back in the right direction.
Absolutely. Thank you, John. And yes, I would love to see it. The Medusa hair falls.
It's in the chat.
Thank you, Evan.
I had additional questions for John. Check for hand raises. All right. Well, thank you very much, John, for that great presentation. All right. So next up, good morning, Zanethia. Go ahead and get set up while I introduce you. So next up, we have Dr. Zanethia Barnett. So Dr. Barnett is a research fisheries biologist for the USDA Forest Service. Barnett's research focuses on understanding factors that impact the structure of aquatic communities and genetic populations, quantifying the effects of human-induced disturbances on aquatic systems, and improving the understanding of life history and distribution of aquatic organisms. Barnett's work in aquatic ecosystems has helped managers conserve numerous aquatic populations by providing a better understanding of how land and water management impact freshwater systems and ways to mitigate negative impacts. Dr. Barnett will speak with us today about genetics in crayfish conservation. Take it away, Dr. Barnett.
Good afternoon or morning, everyone. Today, I'm going to talk to you about integrating genetics into crayfish conservation. So shifting the gears a little away from lamprey to another cool aquatic organism. I'll first take some time to give you a little background on crayfish and their diversity. So there are over 650 species worldwide, with about 400 species in North America. And so over half of the crayfishes in the world reside here in North America. And so we are the hotspot. We are the place to be if you're interested in crayfish diversity, the impacts of crayfishes to ecosystems, and understanding their importance to our ecosystems. Unfortunately, crayfish aren't evenly spread throughout the United States. If you look at this map, I don't have numbers in every state, but as you can see, the Southeast is the hub for crayfish diversity. There are currently actually 100 species in Alabama alone. So a third of the crayfish species that is found within the United States is found in Alabama. And the numbers for the other regions, for the states that's shown, do a really good job of representing the species that's in the other states around them. So there are only few species outside of the southeastern United States. And so a lot of times when people think about crayfishes, if you're talking to the public, they're normally thinking about food. And if you're talking to managers, a lot of times they're thinking about invasives. And but crayfish have a lot more benefits than the negative impacts of invasives and just simply being a food source. They are very important to their environments. They act as ecosystem engineers by processing detritus. They change macrophyte biomass. They manipulate and mobilize substrate. They're a predator of numerous organisms, and they're prey for over 250 different aquatic and terrestrial species. So they play a great role within their ecosystems, and there's been lots of documentations of ecosystems changing when crayfish are left out of those systems, or even through the addition of crayfish in systems. But what we know most about are those invasives and those species that we eat, which is less than probably 5% of the species that we have in the United States. And so while we know that crayfish are very important to the systems, to our aquatic systems, they are actually the second highest species of conservation concern. So over 51% of species within the United States are some type of conservation concern, from presumed extinct to vulnerable. And so although we have lots of species here, they're very important to our systems, we are losing them at an alarming rate. And so lots of funds are currently being put towards crayfish conservation. There's lots of work with the Fish and Wildlife Service, where a number of species were petitioned a few years ago. So work is being done to understand if they should be federally listed or not. And managers are also doing work to conserve the species that are within their drainages and systems that they work in. But most of the work that's being done focuses on richness and abundance. So how many species do we have there? What's the abundance of those species? If we deem it high richness, high abundance, then we say yes, then the system is healthy, the species is doing great, we're all good. There's very few population genetic assessments. So very few assessments that's looking at genetic diversity, fragmentation, movement within populations to truly understand if these populations are actually doing well genetically. And the main two reasons why we don't have those types of studies is because, one, they're expensive, and two, you have to have a special expertise in order to run those genetic analysis. And a lot of times those doing these studies may not have that expertise. So my thought was to try to understand if population genetic and the way we're currently managing richness and abundance are correlated. And one note I have here, that most of the population genetic analysis that are currently being done occur in Europe and Australia. And so in the countries where, in Australia, I think is the second highest country, but where we don't have as great a diversity as the United States, they are currently using these population genetic analyses to conserve their species, but it's not being done abundantly within the U.S. yet. So the goal of this project was to understand the relationship between species level and population level diversity. And so, as you all probably know, there is a hypothesis that species and population level diversity should correlate. So if you have if you're looking at the species genetic diversity correlation, it's a balance between colonization and local extinction. And for a number of species, if the species richness is high, then genetic diversity for the species there is also high. And then you have abundance genetic diversity correlation that looks at environmental conditions that promote or suggest that environmental conditions that promote high cumulative multi-species abundance should also translate into relatively weak effects of drift that drives loss of allelic diversity. So high abundance should correlate to high genetic diversity. And then we have the more individual hypothesis where species richness correlates with cumulative species abundance. So species richness may be enhanced if populations of those species are large and stable. So there's been a number of studies. For a lot of species, these correlations hold up. For some species, they don't, but it hasn't been assessed for crayfishes yet. So we're unsure if just simply doing these species level assessments and using that to judge if a species is doing well or not actually correlates to how a population is doing. And so to assess this, we collected community data, so our species level data from 2 systems in Alabama. And as I stated, Alabama is the highest, has the highest species richness of crayfish within the U.S., with having 100 species and a few that will be named soon coming. So they have high species diversity. We looked at 2 drainages. We kick seine and electrofish for crayfishes in these drainages, and we assessed species richness and abundance. So although we looked at 2 drainages, for the sake of time, I'm going to just talk about one of the drainages. And so in these drainages, there were 2 streams that were impounded and 1 that was unimpounded. So we also looked at the impoundment effects and also took habitat characteristic variables while doing our species level sampling. So for our genetic data, we had 2 focal species. They were both tertiary burrowers. And so this simply means that they're stream dwellers. They all crayfish have the ability to burrow, but these species only burrow during drastic conditions. So if it's a drought or something like that, where they have to burrow to get to water. They live about 3 to 4 years. They mature at about 1 to 2 years, and they occupy small to medium streams. And they both are native to the southeastern U.S. So while these species have numerous things in common, the main thing that they don't have in common is the size of their range. So Faxonius validus has a much smaller range than Faxonius ericksonianus, where Faxonius validus is mainly found in Alabama and Tennessee, and Faxonius ericksonianus is found from West Virginia down to Alabama and Mississippi. So it has a much larger range. But other than that, these species are very similar in their life histories. And then the genetic data collected for these species include the mitochondrial CO1 gene, and then we use ISSR nuclear markers to assess the nuclear gene. So for these assessments, this graph here shows crayfish density. So this looks at the more individual hypothesis. So crayfish density, this is the density of all crayfishes collected within a site. And then species richness. So what was the richness of species at that site? And so here we see that there was no correlation. So we didn't see that, oh, the more abundant crayfishes were, the more species that you would see. We often saw at a few sites that there were definitely dominant species, but richness did not increase just because there were more species there. There was no correlation. So for our species genetic diversity correlation, this is showing Faxonius validus, but we saw similar results for Faxonius ericksonianus. For genetic diversity here shown on the y-axis is nucleotide diversity. And we also look at haplotype diversity, phylogenetic diversity, and proportion of polymorphic loci, and they all had similar results. And again, there was no correlation at all. So we did not see species genetic diversity correlation where you have more species, you have higher number of species, that the genetic diversity actually was higher within these sites. But when we looked at the abundance genetic diversity correlation, and this was actually a surprising result to me, and the genetic diversity measure used here is haplotype diversity, and then we have multi-species abundance, so the abundance of all the species at the site, we saw a negative correlation for Faxonius validus. We only saw it for this one species, but what we saw was that the more abundant the total crayfish population was or community was, the lower genetic diversity that we saw for this species at a site. So that was really interesting in that we only saw that for the one species that has the smaller range, we only saw this negative correlation for that species. So since we didn't see any correlations mainly for most of the correlations, but we did see that negative correlations. We then wanted to understand why didn't we see positive correlations between our species and population level diversity metrics. So we tried to understand if there was some type of habitat characteristics that were driving these diversity metrics in different ways. And the habitat characteristics that we use all were around, we used stream width, which looked at the size of the stream, and then habitat characteristics. So things like how much large woody debris, how much vegetation, the size of substrate, those type characteristics. And so what we saw with that, environmental characteristics affected our species and population level metrics differently. So stream width was positively correlated with genetic diversity and species abundance. So they both correlated positively with stream width, which makes sense. The further you're going downstream, the more input you have from tributaries, the more genetic diversity, the more species, the more abundant species could be. There's more space, there's more habitat. But then when we looked at our other environmental characteristics, they differ based on if you were in an impounded or unimpounded stream. So stream width actually differed with species richness, where in our unimpounded stream, we did see that positive correlation, but in our impounded stream, there was a slightly negative correlation there. And then when we looked at our cumulative multi-species abundance, and we look at large woody debris, we saw a negative correlation in our unimpounded stream. So the more large woody debris you had, the less species abundant, the lower abundance at those sites. But there was no relationship in our impounded stream. And then for substrate, the larger the substrate, the more abundant species were, or the more abundant crayfish were at sites. But the key thing here is that these environmental characteristics differ between impounded and unimpounded streams. And species richness and multi-species abundance were also consistently lower in our fragmented habitats. And so it could be that we need to do additional assessments simply in unimpounded streams, simply in impounded streams, to try to understand the effects of impoundments. And maybe these correlations would hold up if we were only looking at impounded streams. But the reality of it is most of our streams are impounded. And so we will need to be able to understand how impoundments are impacting these correlations, especially if we want to conserve the species in those habitats. And so the impacts to conservation here is that high species richness or high abundance does not correlate to high genetic diversity. That was one of the key findings. And this could even be a negative relationship in some cases. So genetic diversity must be assessed to conserve population. So we can't just simply use our richness and our abundance measures to infer that our populations are doing well. We must begin, when writing our grants, adding in these genetic diversity analysis so we can truly understand and conserve these populations, protect the genetic diversity that they have. And then we also saw that all species are not impacted the same. So we can't go into a site and then choose one species that we think is important and then infer that the other species there are behaving the same way, because different species are being impacted differently. And so these results are published in PeerJ, and if you're interested in reading more about them, the paper is shown there. So now, since we know that we need to understand about our population genetic diversity for our crayfishes, I'm going to jump to a study where I actually do some population genetic assessments to look at a primary burn crayfish. And so we talked about tertiary burrs, which are stream crayfishes. Our primary burrs are crayfishes that do not use surface waters, but use groundwater as their water source. So they can be found in riparian zones, in ag fields, and ditches, in our semi-terrestrial habitats, wetlands, but groundwater is their main water source. And these primary burn crayfishes consist of 15% of the United States crayfish fauna, but 32% of them are imperiled species. So there's a lot of imperilment, a lot that we don't know about these burrowing crayfishes. And one, because they're underground most of the time, and they're actually hard to collect and hard to assess because of that. So the focal species for this study was Distal Canberra's crocori. It's a primary burrowing crayfishes. The IUCN lists it as threatened. It's historically found in Piedmont prairie habitats in South Carolina. And if you think about South Carolina, you likely do not think about prairies, but that was one of the dominant habitats that used to be in the state. And so basically most of all of their habitat is basically gone. And so now we currently find the species in wildlife openings, in roadside ditches, and vernal pools, and fields. So the Forest Service, actually, the National Forest in South Carolina actually contacted me and wanted me to look into, do some work on this genus. And so I want to talk a little about why this genus is important to the Forest Service. So most of the species in this genus are endemic to South Carolina. So there are 5 total species and 4 are endemic to South Carolina. One is found in Georgia, and it only occurs though in the Piedmont regions of South Carolina and Georgia. So the range of multiple species are within the Long King and Inner Reed Ranger districts, which are circled there in green. And so the forest wants to understand how forest management is impacting this genus and so that they can more effectively manage and promote healthy populations. So some things that have happened on the forest is that the forests have cleared open fields to provide wildlife feeding areas and as well as for dove hunting. And what they found is these fields are where we're currently finding these Distal Canberra's crocori populations. There have actually been few collections of this species in forested areas. And so one understanding, or one thing the forest wanted to know, is if how is forest management impacting the populations? Is the forest acting as a barrier to movement between these populations, since we really only find them in these open fields? Do we need to create more spaces for the species? More open spaces for the species. But how is the species doing on the forest? What may be fragmenting these habitats, and how to better conserve them? So the objective of this study was to assess the genetic diversity and structure of distal Canberra's crocodile. I had 2 research questions. Are populations genetically structured, and is genetic structure constrained to drainage pathways? My hypotheses were we predicted that populations will exhibit low levels of genetic connectivity and high levels of genetic structure, and genetic structure will not be aligned to drainages. And so we thought this due to low vigility and decreased suitable habitat. And so we thought with very little suitable habitat, thinking about these open areas, which are far few in between, that there would be low genetic connectivity. And we also thought that drainages, most of the time when we're managing crayfishes, regardless if they're burrowing crayfishes that are in these semi-terrestrial habitats or a stream crayfish, they're managed based on the drainage that they occur in. And so we needed to know if drainage mattered at all for these species, and how should we be structuring the way we manage them. So what you see here is a map of our collections. Each point represents a site where we collected distal Canberra's crocodile. We collected them from 16 sites, 9 drainages, 9 Huck 12 drainages, which is the polygons that you see. And then the site spanned about 80 kilometers. So we also use the mitochondrial CO1 gene to do these population assessments. There were 57 individuals assessed. We found 28 haplotypes and basically pretty high genetic diversity within the populations. So for my first questions, are populations genetically structured? Here we see that same map that shows the sites where we collected crayfish, but the colors now represent genetically distinct populations that were shown through a Samova analysis. So we conducted a Samova analysis, putting in all of our spatial data, all of our genetic CO1 data, and pulled out genetically distinct populations. So there were 5 genetically distinct populations, with 1 large red population in the southern portion of the map, green population near that red population, and then in the northern portion of the map, the northwest portion, we had 3 additional populations. So if we look at these populations in a haplotype network, we see that the red population is not structured. Looks like there could be potentially some recent expansion there. And then the other populations, though, we definitely see structure between those populations. There was only 1 shared haplotype between the populations, so there doesn't look like a lot of movement occurring between those populations. They look pretty isolated, the separate populations. So our population is genetically structured. The northern population was structured. We sampled 2 drainages, and there were 3 distinct populations. So there looked to be low genetic diversity within these populations. The southern populations were not structured. We sampled 7 drainages with 2 distinct populations. There was high diversity within those populations, and it looked like possible recent expansion occurring. So my second question is genetic structure constrained to drainage pathways? And so first we see all the small Huck 12 drainages. And normally for our stream crayfishes, you would see structure based on those drainages. And so our red population spans a number of Huck 12 drainages. And so they aren't structuring these populations. And if we look at our larger drainage, so we have the Savannah River drainage on the left side of the blue line, the Saluda River drainage on the right side of the blue line, those larger river drainages also do not structure our population as we have this red population that's on both sides of those drainages. So, we want to know what is actually structuring these drainages. So we moved on to look at our physiographic regions. And what we definitely see is that we only found distal Canberra's prochloride in the Carolina slate belt. And so I know you probably see a lot of area that it doesn't look like we sampled, because I only show where we collected the species, but we definitely sampled in the southern outer Piedmont region, which is that tannish region and we did not collect any we did not collect many distal Canberra's prochloride from that region. So it looks like this species is aligned to the Carolina slate belt. In the northern portion of the slate belt, you have granite as the soil type. In the southern portion, you have the Carolina terrain. And so all of these soils, though, are large, flat, erosion-resistant areas that retain fine clay sediments during rain events and hold water during dry periods, which creates this perch water table. And if you're a crayfish that needs to dig down to groundwater or to the water table in order to stay moist, having a perch water table is definitely beneficial. And so it seems like these populations are structured based on the geological structure in the area. So next we looked at isolation by distance based on the different, I used the southern, which is that large piece of Carolina slate belt, and then we have that northern small section of Carolina slate belt. So I split those up for the isolation by distance population. And since those populations also split up when we looked at the haplotype network. So when we look here, there was isolation by distance for our southern population. So for that red and green populations, there was isolation by distance, which indicates that there's likely no fragmentation occurring there, because crayfishes or populations, sites that were closer together were more genetically similar, sites that were further apart were more genetically different, which is what we would expect. But when we look at isolation by distance for our northern populations, we don't see isolation by distance, sorry. So we see that crayfish that are close together are sometimes very different, and those that are further apart are less different. And I think the one interesting thing, if we look at our black and our orange populations, the two dots are kind of almost on top of each other. And these two sites were actually on the same ditch, down on different ends of the ditch, and showed up as two separate populations. And so I'm not sure, I didn't walk the full ditch, so there could be some type of pipe or something within these ditch systems, how they're managing the ditch systems, but there were two separate populations found within the same ditch. So we know that the geological area impacted where we found these crayfishes. But another thing that was interesting was looking at land use. And so in the southern range, where we had our red and our green population, it was mostly the Sumter National Forest there, but in our northern range was mostly private lands. So this is another map. And instead of seeing our drainages, here you see land use. So our green represents forests, our yellow represents agricultural lands, and our reds and pinks represent developed lands. So as you can see, that red population is mainly on green, it's mainly on our national forests. But the black and yellow population, those northern populations, are mainly on agricultural lands. And so what we hypothesize here is that forests facilitate gene flow and agricultural lands resist gene flow. So the management and most of the lands up there were in the private area, the northern area, the ag lands, we mainly collected those species out of ditches. And so the management of ditch systems seems to be really important to gene flow. I know in the national forest, they aren't digging out their ditches. They only mow during certain times of the year. So ditch management may be very important to gene flow for these species. So for my second question, is genetic structure constrained to drainage pathways? No, ecoregion and soil types are likely constrained in gene flow. And then within those ecoregions, land use is impacting it, where forest seems to facilitate gene flow and agricultural lands may resist gene flow. So for conservation, my thoughts are that we need to treat the northern and southern sections as two separate units for conservation efforts. For me personally, I need to do some more sampling within that northern range to try to get more data points to truly understand what is occurring within the northern section and what may be causing fragmentation and less gene flow in there. And then trying to develop some best management practice for agricultural lands and ditch management to help conserve the species that live in those areas. And with that, I'd like to thank the Forest Service, University of Mississippi, and West Liberty University for funding and assistance with all of the sampling that occurred for these projects. I will take any questions.
Okay, thank you, Zanethia, for that great presentation. Do we have any questions for Zanethia? All right, looks like Evan has a question. Go ahead, Evan.
Howdy, Zanethia. Great presentation. Pardon my ignorance, but we have two drastically different regions in Idaho and two very different strategies for the ditches and canals. Up north, where we have a lot more rain, they actually use the ditches and canals to drain the water off the ag fields. And so I was just wondering what you were finding in that northern zone. Was it, were they trying to irrigate or were they trying to drain the fields?
So there would be draining, more draining than irrigation. Yeah, trying to get the water off of the land there. And I think that's the key with why I think maybe digging, I'm not sure the time of year, like when they may be digging ditches, but to continue to not have water sitting. So I think during not every year, but during certain years, they may be digging out those ditches to continue to have like good drainage within the systems.
Hi, this is Mari. Can I ask, is the suspicion with the agriculture land that it has generated more habitat fragmentation and isolation than the forested land?
Yes, that's the thought. We actually were thinking it would be the opposite way, where because we haven't found a lot of populations in forested areas that they might not be able to like move through those large forested areas. But it looks like, you know, the agriculture land, so even though they have these open spaces, and you do find populations in the ag fields as well, so they are able to live within these ag areas. But I think it's what, you know, it could be pesticides, herbicide, whatever they're using to manage their lands is not conducive to crayfish and their movement throughout those systems.
Yeah, that's fascinating because I never would have thought crayfish in the forest. Thank you.
Welcome.
All right, we have time for one more question. Anybody else have any questions for Zanethia? Okay, thank you, Zanethia, for that great presentation.
Thank you.
All right, so next up we have Jeremy FiveCrows. Good morning, Jeremy. Take some time to get set up while I introduce you. So last but not least, we've got, I'd like to introduce Jeremy FiveCrows. Jeremy was born and raised on the Nez Perce Indian Reservation and holds a BS in Conservation Biology. He is the Communications Director of Crayfish and has worked there for 26 years. In his role, he manages the organization's external and internal communications, including website and social media presence, media relations, and communications with tribal members, drawing on his scientific background to make complex information accessible to general audiences. His deep connection to the environment, shaped by his childhood experiences in the Nez Perce homeland and time spent in Norway, along with his previous role as editor of the Nez Perce tribal newspaper, informs his work of sharing both scientific research and tribal traditions to foster a broader understanding of tribes' relationships with natural resources. Today, Jeremy will speak with us about the parallels between Indigenous knowledge and genetics and fisheries restoration. Take it away, Jeremy.
All right, thank you. I'm glad to be here. And hello, good morning, everyone. Thanks for the introduction and reiterating that I'm a member of the Nez Perce tribe, which is one of the 4 member tribes of the Columbia River Intertribal Fish Commission that operates out of Portland, Oregon. And today I'll be sharing this is kind of based on an article that was published in Molecular Ecology Resources, first published online back in 2023, and it will actually be in a print version that comes out for the February 2025 print issue on the contributions of Indigenous knowledge in the field of molecular biology. And you can go ahead and scan that code if you want. And I'll put the code up at the end too if you want to go directly to the article about that. And so I participated in writing this with 4 other authors from my organization, including our lead geneticist, Sean Narum, and our senior fisheries geneticist, John Hess, who presented a little bit earlier. So starting off with a bit of background, the Columbia River Intertribal Fish Commission is the fisheries management organization created by the 4 treaty tribes in the middle of the Columbia Basin. You can see where the Columbia Basin is in the North American map at the bottom that's shown in the little brown, in the darker brown. But in the main map are the 4 member tribes. The lighter shade of each is the tribe's ceded territory, and the darker shade is the present-day reservation boundary. And the 4 tribes are the Yakima in Washington State, the Umatilla and Warm Springs tribes in Oregon, and the Nez Perce tribe in Idaho. And together, the ceded lands of these 4 tribes cover 88% of the remaining area still accessible to salmon above Bonneville Dam, which is the first dam on the Columbia River. All 4 tribes also share co-management authorities for Columbia Basin salmon fisheries as guaranteed in each of their treaties with the United States. Crayfish was established in 1977 when the 4 tribes decided to pool their resources to create an organization they could all use and benefit from. So we provide our member tribes with technical assistance, policy development, and scientific research to further the shared goal of putting fish back in the rivers and restoring the watersheds where fish live. The 4 member tribes sometimes say we're all Waikanishpum, which means translates as salmon people, to acknowledge our deep historical and current ties to this fish. We have a deep cultural connection to salmon, which is one of our sacred first foods. You can see on this graphic the 4 first foods, which are salmon, game, roots, and berries. The other foods are included in the first foods. However, these 4 represent the 4 categories of like the aquatic life, land foods, and then the roots and the berries. How these foods have sustained the tribes in our region since time immemorial. And while each is essential, salmon was and continues to be particularly important. Salmon's a significant food source. They provided half the calories tribes consumed historically, and today salmon is still a regular food enjoyed by tribal members. In fact, the current fish consumption rate of our tribes is over 10 times the US average. And in this photo, you can see plates of salmon ready to be served at a feast on the Umatilla Reservation. Salmon's also a central part of our religions. Tribal teachings view the salmon as a gift that sustains life in the region, both human and non-human. In this photo, people have gathered in a longhouse for a ceremony, and after a prayer and a sip of water, salmon is always served first at ceremonies and feasts. Salmon was also the driver of our economies, facilitating trade, driving intertribal relations, interactions, and travel. In this photo, a woman is drying salmon that her family used for both food and trade. Today, commercial fisheries are often the primary means of earning a living for many tribal members. And today, this deep connection also informs our modern restoration and research efforts. CRIPFIC's work is very technical in nature, however, it's guided by tribal leadership that uses the same cultural connection to the salmon that informs all their other activities. This outlook of incorporating traditional Indigenous knowledge and Western science has taken decades to develop and fine-tune. When CRIPFIC was first established in 1977, scientific research was largely conducted through a lens Elders Albert and Radina Marshall labeled as one-eyed, meaning it viewed Western science as the only valid knowledge system. So when tribes would cite data or information from oral histories or other sources of traditional knowledge, scientists would often discount or outright dismiss it. Over the subsequent years, as keepers of other knowledge systems, such as Indigenous knowledge, received more attention, Western science was exposed to it more and more. Often, however, scientists wouldn't value the information unless Western science confirmed it. And when it was confirmed, it was just assimilated into the one-eyed seeing paradigm, resulting in it losing some of its context, nuance, or value. Today, there's a growing call to operate where both worldviews coexist in tandem, to use both in parallel to produce a mutual understanding between tribal and non-tribal knowledge systems. Marshall explained it as to see from one eye with the strengths of Indigenous ways of knowing, and to see from the other eye with the strengths of Western ways of knowing, and to use both of these eyes together for the benefit of all. So this is the model that CRIPFIC aims for in our work. So by this two-eyed seeing method, we kind of value both systems as valid ways to view the world, as well as take advantage of the unique attributes that each knowledge system provides. In the example on this slide, the overlap between Indigenous knowledge and genetics highlights the nature of biological information being passed over generations that lie at the heart of both circles. And not only is the overlap a symbolic connection, the two circles highlight the very real data that can be gathered. Because knowing, for example, both phylogenies as well as environmental timing cues gives us a richer and more complete understanding of the world. I'll pause here to point out that the general public's ignorance of what traditional ecological knowledge even is can be pretty harmful. I'm sure everyone here on this webinar is familiar with the common trope of like magical Native American powers coming from traditional spirituality, or because they're close to nature that civilized races don't have. But if people act in bad faith, paint traditional ecological knowledge and Indigenous knowledge as just Hollywood-style mysticism, rather than actual knowledge and wisdom, it can be used to both discount tribes as well as the efforts for agencies and departments to actually implement Indigenous knowledge in their operations. So you know, in order to kind of dispel some of that, I'm going to give some specific examples of how we have melded Indigenous knowledge with Western science in by highlighting 3 projects that either we've done or our member tribes have done in our efforts to protect and restore salmon and other fish species in the Columbia Basin. The first is sustainable harvest management. Management of Columbia Basin fisheries is extremely complex. Managers have to take into consideration issues such as run forecasting, minimizing impacts to ESA-listed runs, ensuring all parties receive their fair share. With so many competing interests and parameters, decision makers rely on good science and data to manage the fishery appropriately and sustainably. In this photo, Yakima fisher Leonard Dave is fishing from a scaffold on the Klickitat River, which is a tributary of the Columbia River. And fishers like Leonard count on these fisheries to feed their families and communities. So tribes feel a really deep responsibility to manage these fisheries in a way that ensures a harvest now as well as for future generations. So this graphic from our article shows several of the aspects of harvest management issues as kind of puzzle pieces, with pieces that each play a role in providing needed and useful information or data. The tan pieces represent indigenous knowledge, and the blue represents Western science. Considered together, they guide our efforts and understanding of a given topic. Now, I'm not going to go through like each item, but I will kind of just zoom in on kind of one example of how this works. Is we see how examples of information gathered from tribal fishers and elders that documenting reports of historical abundance or observed changes in the run. So this information draws from decades of firsthand experience and observations that fishers have personally witnessed, as well as oral histories documenting past information that can extend back generations. We also conduct surveys and genetic analysis using more traditional scientific tools. Let's see if this works. Oh yeah. So at our facility at Bonneville Dam, Crayfish technicians collect fish tissue from each sampled fish to be sent to our Hagerman Fish Genetics Lab for sequencing and analysis. So data like this, coupled with the traditional information, provides a richer and more detailed estimate of historic and current salmon populations that forecasters and fisheries managers can use to guide their decision making. My next example is the tribal hatchery supplementation programs that are working to rebuild struggling salmon and steelhead in all their traditional fishing locations. In this photo, a Nez Perce fisheries technician is releasing salmon smolts by hand into a remote Idaho creek. Some of the science of supplementation is still being studied, and we're constantly fine-tuning or expanding our understanding of the techniques, optimizing hatchery operations we use, and analyzing impacts of the programs to ensure the results are effective and also scientifically sound. With an overall goal of boosting salmon abundance and reproduction, tribes are melding the two knowledge systems to guide the many facets of this work to ensure the efforts are effectively rebuilding populations, monitoring any impacts to genetic diversity of the population, and also promote the rebuilding in ecological and culturally important locations. For example, when identifying potential sites for supplementation programs, we gather information such as locations of historical fishing camps, which indicates areas of strong salmon runs, or conduct interviews to collect knowledge of fish behavior gathered from fishers who have decades of firsthand experience, as well as information passed down from their elders. Then we can add in research that analyzes factors such as phenotypic diversity or metapopulation structure. We then use all this information to determine restoration locations that provide the maximum benefit and efficacy to both the fish population, the ecosystem function, as well as cultural benefit. So like in 2017, after years of work compiling data, research, and histories, the Nez Perce tribe successfully released coho salmon into the Lost Steen River in northeastern Oregon. Coho had been extirpated in this river for over 40 years, and the release site was near Chief Joseph's, the leader of the Nez Perce's winter camp, which marked also an important cultural connection to the release as well. And the last example is the tribe-specific lamprey location program. And actually, I just love that, like the fact that it's like I like the crayfish are cool, but I like knowing that like we're having not only began the series with lamprey, but we're also kind of like ending with an example of lamprey, just because I'll get on with the connection I have with lamprey too. But anyway, this last example is the tribe-specific lamprey translocation program. And this effort assists dwindling or struggling lamprey populations in the region. And John Hess had discussed some of the nuances of this as well. But lamprey have difficulty passing dam fish ladders that were designed for salmon. And as a result, lamprey have trouble reaching upriver areas that increase the more dams they have to pass. For example, lamprey attempting to migrate to a stream in Idaho have to pass 8 fish ladders that weren't even designed for their unique needs. So to address this, in 2007, the tribes began collecting lamprey at lower river dams to transport on trucks to be released in upper tributaries. It's like a stopgap measure to aid lamprey. But for the time being, it's a way to increase the numbers of spawning lamprey throughout the region. But also because it's kind of a relatively new technique, it's critical to gather more information to evaluate the effectiveness of the program and also just to fine tune it. So both the efficacy of the effort and the impact is important to determine, in our effort to protect and restore these ancient fish. So adding to this significance is the difficulty of the fact that lamprey haven't been studied as extensively as, like, for example, salmon. So part of the work actually also involves just advancing our understanding of lamprey biology in the first place. Again, this involves gathering information about behavior, habitat preferences, historical run timing with genetic analysis of adaptive traits and life histories. This results not only in furthering of general scientific knowledge, but it has also confirmed great news in the effort to restore lamprey, which actually John mentioned this in his presentation too. But when our genetics lab discovered the first adult offspring returning to the Columbia River that was the offspring of parents that had been translocated themselves. And what made this discovery even more significant to me personally was that the parents of that lamprey had been translocated and released by my father, Elmer Crow, and he's the person that's on the right there leaning over to put some lamprey into the river, that he had released himself back in 2007. And his kind of life's work revolved around lamprey, out of both like kind of like not only a cultural connection, but also he just had this total like contagious enthusiasm for lamprey. So much so that his name is Elmer, but he was often nicknamed Eelmer. But he passed away in 2013. So to have a lamprey coming back even now from his direct effort shows that his work is still making that big of a difference. This actually really isn't part of the original presentation, but I had to throw in this photo of a crayfish eating a lamprey ammoset for Zanethia. But it's also not only is just kind of acknowledging that, but also it's just a good reminder that everything plays a role in a healthy ecosystem. And like you can see one, they're not like just kind of this parts that you can pull out and study in just by themselves. You have to see this as a kind of a big picture. So here, this graphic is a representation of the Nez Perce seasonal round, but it shows this. It's not only the major foods and gathering times that the Nez Perce depended on, but it highlights that interconnectedness of the ecosystem and how things lead into each other or that are connected with each other in the interplay with the seasons. And so with two-eyed seeing, it's possible to see both the interconnected whole, like represented here, as well as the individual parts. And so the three examples I shared of how seeing the world through both lenses hopefully kind of highlights how this enriches the research and outcomes of all of our efforts, not just tribal sciences working on this, but like everybody. And so as this recognition and relationship building occurs, hopefully we can all broaden our toolboxes to continue this shared work of kind of expanding human knowledge of the world all around us. And with that, I thank you and open any questions.
All right, thank you, Jeremy, for that presentation. Great presentation. I think Eelmer would be proud. All right, I'm not seeing any questions in the chat. Do we have any questions online?
I think Sharon might have had her hand raised if she wants to go first.
Yeah, hi. Thanks, Jeremy. That is a fantastic presentation. Love that presentation. I would like to know a little more about some other projects besides genetics. Are you aware of, maybe not even at CRITFIC especially, but among the CRITFIC member tribes, where traditional ecological knowledge and the two-eyed seeing sort of framework is being applied to research projects? But are you aware of other examples or ways that it's being done?
Well, I've been, these examples are some of the ones that kind of like, kind of highlighting these of the work of CRITFIC, but knowing that there's the President Biden had listed in an executive order about involving tribes in land use management had been a kind of a growing effort, and so there was a lot of effort by different federal agencies to of like what that looked like, and that's why I kind of threw in that Simpsons one of like how that is kind of painted as like it's not just kind of like this mysticism type thing, and I think that's where we're seeing things like the traditional burns that the in land management that's we're seeing growing use of there. Some of the bison reintroduction efforts that are happening throughout the plains is also kind of informed by those by that knowledge, and also I would probably turn over for like actually specific research projects that have been going on to if Sean or if John Hess have any specific examples beyond what we'd done. But yeah, we kind of like have seen that growing awareness.
Thanks for that. Yeah, I'm aware of too, in Canada, where there are some land management projects being done, restoration even, and other sorts of things being done by First Nations people, such as ways that they manage shellfish harvesting, are really being considered as the primary source of information, with some Western science being incorporated as a secondary or after, sort of after the fact introduction to what's happening on the ground. And I think that that's a pretty good model to see. Yeah, Sean, I don't know if you're aware of other instances.
Looks like we have a question from Evan Brown from the chat. Is there an online library from which we may access, if not linked to Indigenous knowledge?
Yeah, so this kind of goes to like I think there's been a lot of reticence of tribal members wanting to share traditional knowledge because it's kind of been exploited in the past, dismissed, or kind of monetized by the people. So I think that I've never heard of like kind of like an online repository of that. But it also kind of speaks to the idea of that this knowledge is like it's so place-based, and so if you're working in an area that it's kind of to be able to build personal relationships with those knowledge holders, and kind of come from a basis of trust that for it to be shared, and then kind of be guided by that knowledge. One, knowing that how it's going to, in terms of how it's going to be used and not exploited, and then also acknowledged in the sharing of that. So it's kind of like in that way, kind of like a point of like humility there of how it's used, as well as kind of the enriching part of the relationship building to do the research, do the interactions, to take advantage of that.
Okay, any additional questions for Jeremy? All right, so we have about 6 minutes left before our end time. If we'd like to transition to general questions for any of the speakers, I'd just like to thank Jeremy, Zanethia, John, and Kellie all for great presentations today. Really informative, and I've learned a lot. Yeah, so does anybody have any additional questions for any of our presenters today? I guess I'll start off with a question for John. So, John, if I understand correctly, it looked like the SARS or the JARS to Bonneville were based off of sample collections from the SMP data that you received from Fish Passage Center. if I'm reading that correctly. And so given that lamprey are kind of notorious escape artists from these bypass facilities and the holding tanks, were you able to account for a potential underestimate of these collection numbers in the daily samples?
Yeah, that's a great question. And no, we didn't take any methods to account for the escaping lampreys from those traps. But what we use that measure for is to build kind of we were hoping that it would at least be a consistent relationship, that if everything kind of happened in a very similar way and that, escaping wasn't, more prevalent one year than the next, that we could just build our relationship between our estimate of abundance using that method and then the adult returns at Bonneville. And as long as those relationships stay the same, that we would see our trend in juvenile abundance would increase, and then it would reflect an increase in adults in future years. And so, yeah, as long as those relationships were fairly consistent, that's how the model kind of held together. But in reality, the true estimate of abundance is likely a lot higher, than what we were able to estimate based on that issue that you mentioned. If there's lots of fish that were escaping, then we weren't counting very many, and that we were missing a lot of those numbers. So yeah, I would expect that if we could account for that, it would just make those juvenile numbers much larger, but they should be larger proportionally across those years.
Thank you, Jeremy, or excuse me, John. Hey, I'm not seeing any additional questions in the chat.
Yeah, thanks, Alan. We have just 3 minutes left in this session, but I wanted to thank everyone who presented today. Great presentations. Also, check the chat. There's some really great links to papers in the chat from Sean and Zanethia and Jeremy. And after this, I will be emailing everyone who attended, and those will provide, I'll provide a link to the recording here once that's all set up, and feel free to share it amongst your colleagues and whatnot. But I wanted to give a big thank you to the ETIS Planning Committee. As you can see, there's a whole bunch of us, and it's taken, a fair bit of time to get this all together, but it's been going quite well. And then I just wanted to let you all know about next week's presentation. It's the last session in this webinar series. It's going to focus on remote sensing, and we have 3 presentations. They're going to be around 30 minutes each, and then we'll have questions and answers for all of those. So, tune in next week, same time, and you can find all the information on pnamp.org as well in the on the Emerging Technologies Information Sessions project page. So yeah, hope to see you guys next week, and thanks again to everyone. And yeah, have a great rest of your day.